I'm going to briefly talk about ovarian cancer staging and how it's staged and how it pertains to our work, and perhaps Professor Hacker will talk a bit more about that. But basically, the way it works is it's staged as early stage or late stage based on how much the cancer has spread throughout the body. So in the early stage cases, ovarian cancer is limited to the ovary or within the pelvic region. And these women are treated with surgery and perhaps with chemotherapy, and in general do quite well. The late stage women, which cause a majority of the mortality from the disease, the cancer actually spreads throughout the what's called peritoneal cavity, which is basically the abdomen and all the organs inside. And you can see that it spreads quite far out. And in the stage 4 cases, the late stage, it even can spread as far as the liver and the lungs. And you can imagine that this is not easy to remove during surgery. And these are the women who tend to have a worse mortality and don't respond as well to chemotherapy. So the problem is that a majority of the women are the ones that are diagnosed at this late stage. So the yellow bars show the percentage of women diagnosed at the different stages, and the blue bars show the survival at those stages. So you can see that women diagnosed at early stage have a very good survival of 90% or so, but only 25% of women are diagnosed at this early stage. Whereas the women who are diagnosed at late stage have a survival of 20 to 25%, and that's a majority of the women. So the goal for us at the Ovarian Cancer Research Group here is to shift diagnosis this way. If we can start detecting these women earlier, we have a better chance of improving prognosis. So we have 2 approaches. The main project of the group, which is what was the founding project of the group, which is what Rob and Neville kind of brought together, is to look for ways to detect ovarian cancer early. And we're using a special alteration of DNA, and I'll talk about that in a little bit. The other project, which is a project I was working on in my graduate studies and in my postdoctoral studies, which is what I brought from the US, is to try and identify other ways to treat these women. We know that the standard chemotherapy we have now clearly isn't effective enough. Can we target other parts of these tumors and help treat the cancer better? So chemotherapy resistance is a problem not just in ovarian cancer, but in many types of cancers. And this is kind of a simplified version of what happens in a tumor, but tumor consists of a bunch of cells, and a majority of these cells can get attacked by chemotherapy and killed by chemotherapy. So that's great. What happens is, within this mass of cells, there are going to be a few that have resistance to chemotherapy. And one of the main mechanisms of resistance are these pumps that are located on the cell, and they are responsible for pumping out the drug. So we're treating the tumor with the drug, and then the tumor is pumping it out. It's not actually going to kill the cell. And resistant cells have an increase in this pump. So this is one example of what happens. So we have a tumor, we have a few resistant cells, we treat with chemotherapy, and the non-resistant cells die. But you see that the resistant cells with the high increase of the pump survive. And then they grow out into a whole new tumor, and this new tumor is resistant to chemotherapy. So no matter how much we give, it's just going to pump it out. So this is the main problem with chemotherapy resistance. So the idea is, well, we know that tumor cells are able to avoid chemotherapy treatment. Can we do something else to treat these tumors? So what I'm studying, or what I studied as a postdoc and brought here, is what's called the tumor microenvironment. If this is a normal ovary here, the ovary consists of a layer of epithelial cells that surround kind of a supporting network of other cells. And this supporting network of cells helps feed the epithelial cells. It keeps it regulated. It nourishes it. What happens in a tumor is you get an outgrowth of these epithelial cells, and you see a kind of increased dependency on this microenvironment. So you see an increase in factors that help stimulate the growth of this tumor. That's how the tumor grows. That's how the tumor spreads. So the thought is, if this tumor is dependent on this microenvironment, can we target this microenvironment and basically cut the tumor off, starve it to death? And that's the idea of the project that we're working on. So the question is, how can we specifically look at the cells in the microenvironment and not the cells in the tumor? So when I first started this project, gosh, I guess in 2006, they had just relatively recently developed a process called microdissection, which allows you to actually pinpoint specific cells in a tumor. So the idea is, we don't want to study this cell, we want to study this cell. So microdissection allows you to do that. There's basically a cap that has a laser that heats up specific cells of interest, and then you can isolate them specifically. Now this, back in that time, was a very hot topic because we know tumors are made up of so many different cell types. This is a way to study specifically what's happening in different types. So we're able to isolate the microenvironment, the cells from the microenvironment, and look at the genes in those cells and say, okay, what genes are in these cells that we can target with new drugs? So this is an example of kind of the output we get with these kinds of studies. These are samples from women with ovarian tumors. So this is actual ovarian tumor tissue, and this is women who donate healthy women who don't have ovarian cancer, who donate their ovaries when they're being removed for other unrelated reasons. So then we can look at hundreds of thousands of genes in these cells and figure out what's different between them. So this is just an example of some of these genes that are different in the normal ovaries versus the ovarian tumors. And the idea is, can we actually target any of these genes specifically? So one of these genes is one that I chose to focus on, which excited me because this gene actually had been found to also be present in pancreatic cancer. And there was a company that already formed a drug against this gene. So they had already put it through early phase clinical trials, so I thought, well, let's test it in ovarian cancer as well. It looks promising. So what we were testing, really, as I mentioned before, ovarian cancer, the biggest problem with ovarian cancer in terms of treating it is the fact that it spreads into the peritoneal tissue, in the peritoneal area. So what we wanted to do was see whether we can prevent this from happening. If we can prevent this from happening, we can keep the tumor at bay. So we need to be able to study this in the lab. Unfortunately, we can't at this point do these kinds of experiments on humans, so we need to study this in the lab. And Kim Jones, who's a postdoc in the group, came up with a really nice way to study this. Mice have peritoneal tissue as well. And when you do ovarian cancer studies in mice, you also see a spread in the peritoneal tissue as you do it in humans. So it's a really nice comparison. What we can do is actually take that tissue from mice. We have ovarian cancer cells that we've taken from tumors that we can actually grow, and we add it to the tissue, and we let the cells stick. We know that these cells stick to that tissue. That's what they do. We can then take the tissue and look at it under a microscope to see what cells have stuck and what cells have not stuck. So this is an example of what that looks like. So if we take the tissue and we take the cells, these little green dots are all individual cancer cells. So you can see that without any treatment or any effect by us, that these cells stick to this membrane. Now what's interesting is, remember I mentioned that gene that's expressed in tumors and not in normals. When we add that protein, you see that you have more sticking. So what we know is that this protein actually drives that sticking to the tissue, and that's exactly what we want to prevent. Well, the drug against this gene, it's called FG3019, that's just what the company names it. If we add that, you see we prevent that. You see much less green than you do here. Now this is one study, this is done in mice. Clearly this is not indicative of what's necessarily going to happen, but this is a good starting point for us to say we know that this gene does something to increase our tumors, and we know that this drug helps prevent that. So now we just need to follow up on these types of studies, do them in actual live mice, and then eventually, if those are positive, we can actually take these to early trials in women. So that's kind of one project. The second project, as I mentioned, which is the main focus of our group, is this early detection project. And the idea is, can we find a way to detect ovarian cancer earlier. We want to shift that curve so everyone who's diagnosed is diagnosed at an early stage and can be treated properly. And we want to do this by looking for a blood-based test, a non-invasive test. It's cheap, it's easy, and it's a way to make it easier to actually diagnose this disease. So what we're doing is we're looking at specific alterations in DNA called DNA methylation. DNA methylation is a natural occurring process. It happens in our bodies all the time, and it's a way to regulate which genes are turned on and which genes are turned off. So it's really important, particularly, let's say, in development, when your hair develops, when your eyes develop, when your limbs develop. When a gene is methylated, it's turned off, and when it's not methylated, it's on. So if this is an example of a gene, and all these little bars are what are called CpG islands, which are basically places in DNA that can be methylated. So here's a CpG island without any methylation, and this gene is turned on. When that gene is methylated, it's turned off. Now what we know happens in cancer is we see DNA methylation happen when it's not supposed to. So we see genes that are methylated and genes that are turned off, and this is what causes your cancer cells to grow and divide out of control and to spread. So what we thought is, well, we know DNA methylation happens in cancer, and we know it happens early in cancer. Can we detect these methylation changes early enough that we can predict, okay, this person is getting cancer, and cut it off before it happens? So, as I mentioned, we're looking for a blood test, and so we know that there is actually DNA, free circulating DNA in our blood. We have everyone has a certain amount of it, but what's interesting is that in patients who have different diseases, including cancer, this amount of DNA floating in the blood is actually increased. So great, can we actually detect free circulating DNA in the blood? Can we look at these methylation changes and use that as a test? So we, with our relationship with Professor Hacker and the Gynecologic Cancer Center, we were able to actually recruit women to donate blood before they were having surgery. These are women who were diagnosed with ovarian cancer. We also had healthy volunteers from the hospital, as well as here at the Garvan and St. Vincent's and the community, healthy volunteers who donated their blood as well. And we basically were able to purify the blood and check for DNA methylation between women with cancer and healthy women. And again, this is what the outputs look like, and they're quite complicated, but I just wanted to give you an idea of what it looks like. So if this is a map of a chromosome, which is DNA we get out of the blood, and this little red line here refers to this area that we're looking at here. Okay. These 6 purple splotches are our patient samples, and these 2 greens, which you actually can't see, are our normal volunteers. So if we look at this particular area, what we see is these peaks refer to methylation that's happening in that spot. And you can see in the 6 patients, we see methylation. In these 2 patients, we don't. So the idea is perhaps this is one of many regions of a DNA where you see difference in methylation between patients with cancer and women without cancer. Now, this is one area of one chromosome. We have hundreds of thousands of these for each individual person. So it involves a lot of analysis and a lot of literally looking through these pictures to find what's different. But that's basically what we're doing. We were able to show that we can see methylation in DNA and blood. We can see differences between patients and healthy volunteers. And now the question is, which of these actually drive the cancer? And so what we'll do is we'll put together a list of these regions and then go and actually test them in women who are at high risk for women who are at high risk for cancer to see whether or not they're going to develop cancer. So I'll skip that. That's where we are now. First of all, I'd like to thank you all very much for your attention. All of this work is being done with the people who are the researchers in the ovarian cancer group, in collaboration with Neville Hacker and the Gynecologic Cancer Center at Royal Hospital for Women in Randwick. We've had a lot of support throughout the years from the Go Research Fund and Cancer Institute New South Wales and Cancer Australia, and of course there have been many very generous donors here through the Garvan and the community who have also contributed to this project. So with that, thank you for your attention. And
thanks very much, Rob, for that kind introduction. I'd like to give you just a brief clinical overview of ovarian cancer. So I'm going to firstly say what is ovarian cancer, what are we talking about when we talk about ovarian cancer, what's the incidence of the disease in Australia, what causes it, how do we prevent it, how do we screen for it, what are the symptoms, how do we diagnose it, how do we treat it, and I want to finish with a case history which will illustrate a lot of these points. Firstly, what is it? Well, what we're talking about with ovarian cancer are three different basic types, and the commonest is what's called epithelial ovarian cancer. That arises from the surface epithelium of the ovary, and this represents about 90% of cases, and that's what we're talking about, that's what Goli was talking about, and that's what we are generally referring to when we talk about ovarian cancer. However, there's an interesting group of tumours called germ cell tumours, and these occur in young women, sometimes in teenage girls, women in their 20s, and these have been one of the real success stories of ovarian cancer because they're highly sensitive to chemotherapy. They used to be almost uniformly fatal, but now they're very sensitive to chemotherapy, and even when they're spread around the body, they can usually be cured. They arise from the egg or the ova. And then there's another group of tumours which arise from this specialised stroma here around the follicle. Examples of that are granulosa cell tumours, and these are usually hormone-producing tumours. That they generally have quite a good prognosis, although they can be lethal. This is the incidence of cancer in Australian women. The latest report from the Australian Institute of Health and Welfare. And you can see, of course, that breast cancer dominates the situation with 12,000 cases a year. It represents about 27% of new cases and 15% of cancer deaths in women. Cancer of the ovary is about 1,200 cases, 2.7% of new cases, but 5.2% of deaths. Apart from lung cancer, it's the only one that's sort of overly represented in cancer deaths compared to cancer incidence. What causes cancer? What causes ovarian? What causes any cancer? Well, we now know that cancer is a genetic disease. It's caused by mutations, changes, which we call mutations in these cells. And these mutations remove the normal control mechanism that the cell has over proliferation. So the cell rapidly divides out of control. 10% of the cancers are actually hereditary. And that means you can inherit them from your parents. And the predominant genes involved there are the BRCA1 and the BRCA2 genes, which also are associated with breast cancer. The bowel cancer genes also increase the risk of breast cancer. And these genes were only relatively recently identified, the BRCA1 in 1994 and the BRCA2 in 1995. And the risk that this confers, the BRCA1 is about 40% of ovarian cancer in the woman's lifetime, and then the BRCA2 about 20%. Fortunately, the ovarian cancers that these genes occur usually don't occur until the 40s and 50s. So it's possible for the woman to have her children, and then we can remove the ovaries and prevent the development of the cancer. So there are some epidemiological factors that increase your risk of having ovarian cancer. And these are things like low parity, early onset of periods, late menopause. These are thought to be possibly related to this concept of incessant ovulation. This seems to be counterintuitive. You'd think that the ovaries are supposed to ovulate, that excessive ovulation wouldn't be causing cancer. But possibly the fact that in the normal animal kingdom, when the female comes of heat, she's usually impregnated by the male and she's pregnant, and so there's not this incessant ovulation that you get in the human species. And this would be consistent with low parity and late menopause and early onset of periods. Long talcum use is thought possibly to be relevant, high-fat diet, and long-term estrogen replacement therapy. To decrease the risk, the oral contraceptive use is important, and this again would be consistent with this concept of suppressing ovulation. Also, hysterectomy or tubal ligation, and that would be consistent with the idea that maybe something is ascending from below, talcum powder, whatever. Red wine, there's some good news. But the best way to prevent it, of course, is to prophylactically remove the ovaries and tubes if there's any significant risk. And over the past 15 years, an increasing number of women with these BRCA mutations that have been identified are having their ovaries removed. In fact, they create quite a lot of space on the operating theatre timetable, and it's getting difficult to get routine cases on because of the number of these procedures that are being done. But interestingly, between 1997 and 2006, the age standardised incidence rate for ovarian cancer decreased in Australia by 25% in this group of women aged 50 to 69. And I imagine that all these prophylactic operations are starting to have an impact. Now, this was an interesting paper out of Stanford 2 or 3 years ago, where they looked at all of these women who had had prophylactic removals. They looked at 8 studies. There was a total of 406 patients. And in about 8% of these patients, the cancer was already present in the ovary or in the tube. And certainly this is an experience that we've had on several occasions where when you get the ovary out, you think you're doing it prophylactically, but there's already an early cancer present. And from this they were able to conclude that these cancers, serous cancers, which are the commonest type and the type that are usually inherited, spend an average of at least 4 years in the pre-cancer or early stage disease, and probably 1 year as advanced disease before they become clinically apparent. And I think this is good news in terms of the possibilities for detecting a screening test. A lot of people have been pessimistic that we're not going to be able to develop a screening test, that these cancers are going to be spread before you can possibly diagnose them. But I think this evidence that we have from these prophylactic removals suggests that if we persevere, it probably will be possible. At the present time, there's no effective screening. The best marker we have is the CA125, and that's a very good marker for following patients on treatment. But unfortunately, there's a lot of false positives with this disease, particularly in premenopausal women who have conditions such as endometriosis, fibroids, pelvic inflammatory disease. All of these things and many others will put up the CA125 level. For patients who have a strong family history, or for women with a strong family history, or proven hereditary ovarian cancer syndrome, a BRCA-positive patient, we recommend a transvaginal ultrasound every 6 months, but unfortunately that's not always effective. We've seen patients even under screening who come in with advanced disease. So how do you diagnose it? Well, in the absence of a screening test, we have to rely on symptoms, and the symptoms, unfortunately, are very vague. Many patients go to their doctor with a short history of abdominal swelling, abdominal distension, and they've already got a lot of fluid there, what we call ascites, and they have advanced ovarian cancer. So the diagnosis ultimately relies on an operation. We looked at the symptoms of ovarian cancer at the Royal Hospital for Women 3 or 4 years ago, and the commonest symptom was abdominal pain, followed by abdominal swelling or abdominal bloating. Bowel symptoms are common: nausea, indigestion, loss of appetite, constipation. Abnormal vaginal bleeding is sometimes a symptom. Postmenopausal bleeding, irregular periods, intermenstrual bleeding. Urinary symptoms, frequency, urgency. Not surprising, because the ovarian mass is pressing on the bladder probably. And lethargy is a fairly common symptom, often mentioned almost en passant, that they've had this significant tiredness recently. But the interesting thing in that study that we did was that 70% of the patients with both early and late stage disease had the diagnosis made within 3 months of the onset of symptoms. So it's not that the doctor is missing the diagnosis. It all relates to the biology of the tumour. If you've got one of these aggressive cancers, it's going to spread before it produces symptoms. But as I showed you earlier, I think we can be optimistic that even those aggressive cancers do spend a reasonable period of time when they're not clinically apparent. You can't pick them up on ultrasound, but we probably will be able to pick them up on a blood test eventually. Now, early cancers, as Goli has said, are those that are confined to the ovary or to the pelvis, and many of those patients can be treated just by surgically removing the ovarian mass. But what we do with those patients is what's called surgical staging. We need to find out whether the cancer has spread, and the common sites it spreads to are the lymph nodes, sitting around these big blood vessels here, and to the omentum, which is this fatty pad here that hangs down from the stomach. Many of those patients, as I said, that have disease genuinely confined to the ovary can be cured with surgery alone and don't need chemotherapy. Unfortunately, the more common situation is the advanced disease, and in these patients we have to undertake what's called surgical debulking, that is, trying to remove the bulk of the cancer, and then they get usually 6 cycles of carboplatin and Taxol. Sometimes we give this 2 or 3 cycles of this treatment before we operate. That's called neoadjuvant chemotherapy, and we like to do that particularly in women who have big effusions, particularly fluid on the chest, because these patients do much better. 1 or 2 cycles of treatment will usually clear this up, and then the post-operative recovery is much quicker. This is a typical sort of situation with a patient with advanced ovarian cancer. You open the abdomen and you see this big mass of tumour here. This is the cancer here. Here's the bowel. And after you remove it, you see all the normal small bowel and this part of bowel left here. So this is what we call debulking the cancer, getting down to very small residual disease. Occasionally, this disease in the omentum will go right across to the spleen, and the spleen will be taken out as part of that. So how do we follow these patients then after they've had their surgery, after they've had their chemotherapy? About 75% of these women with advanced ovarian cancer will be in what we call a complete clinical remission. That doesn't mean they're cured, but it means that for the moment all the scans will be normal and the CA125 levels will be normal. So we follow these patients every 3 or 4 months for the first couple of years, and about every 6 months until 10 years, and we will, certainly the advanced patients, and we do see a few advanced patients who get to 10 years without any recurrence, not very many, but we certainly see a few, and we would continue to follow those patients at least once a year. We get the CA125 titers before we see them. We don't get any routine x-rays or scans because the CA125 titer will reflect any recurrent disease quite effectively. But if the CA125 starts to rise, then we will order a CAT scan or a PET scan. And then there are various other treatment options that we can undertake. There may be need for further surgery, chemotherapy, radiation, hormonal therapy, or these days targeted or gene therapy that Goli was referring to. So these are the sort of survival data, and it's probably not quite as pessimistic as Goli suggested, but it's not much better. You can see these are figures going back to 1962. This is the improvement in survival for stage 1. This is the overall survival, and this is the survival for patients with advanced disease. And things did improve up until about the mid-90s, and they seem to have plateaued at that point. But I think that they probably will start to improve again with some of these gene therapies. Why have we got this improvement, modest though it is? Well, certainly I think the subspecialisation in gynecologic oncology has been an important factor. Patients now can go to properly trained cancer surgeons. In the past, you went to your local obstetrician and gynecologist who probably delivered a baby at midnight and then had to face one of these big operations the next day. And so naturally, it was a fairly substandard operation that was done. But with the subspecialisation, doctors are now trained to do these more extensive procedures. Better chemotherapy, particularly since the introduction of the platinum compounds in the early 80s, they have made a difference and been also responsible for improving things. And I think the centralisation of care, referring patients to cancer centres rather than treating them in various hospitals around the state. But to improve, to get major improvements, we're going to have a screening test which will allow early diagnosis, something that can be applied to the population of women, a blood test, a urine test, or something of that nature, where these tumours can be diagnosed when they're still confined to the ovary. Now, I just want to finish with this case history. This is a patient, 68, who presented in March 2004, typical abdominal swelling for 3 months and a change in bowel habits for 1 month. We did a colonoscopy to make sure she didn't have bowel cancer, but that was clear. She had a very high CA125 titer. CAT scan showed that she had bilateral ovarian masses. She had quite a big mass in the omentum. She had ascites, and she had lymph nodes in both groins. Her mother had had breast cancer at the age of 44, and a maternal grandmother had ovarian cancer. So you could predict that this woman had hereditary ovarian cancer, and in fact when we did the testing she was a BRCA1 mutation carrier. So we did the bulking operation that I described, and she had a stage 4B, the most advanced type of ovarian cancer. She was slow to respond to the carboplatin and Taxol. CA125 titers fell to normal after about 6 cycles, so we usually give 2 cycles beyond normal titers, and she had a total of 8. She had a final chemotherapy October 2004. By September the following year, the titers were going up again, but a CT scan at that stage was normal. So we treated her with tamoxifen, which is an anti-estrogen type of a drug, which works about 30% of the time and is quite a useful thing in this situation. Her CA125 stabilised for a few months until May 2006, when they started to rise again. She was then commenced on this drug, pazopanib, which is an inhibitor of new blood vessels. This is one of these gene therapies that Goli was talking about. In order for the cancer to grow, it has to be able to produce new blood vessels to sustain the growth. And she had an excellent response to this treatment. In fact, the response lasted 4 years. And it's a tablet, you take the tablet, it's pretty well tolerated, there's very few side effects. Then the markers started to go up again, so she was changed to a different type of chemotherapy, which this time a BRCA inhibitor. Because she had the BRCA mutation, this gene therapy here works on these type of patients. Again, she had a good response, which lasted until October 2011. The titers started to rise. That was 7.5 years since diagnosis, and we retreated her with carboplatin. And she went into another remission and has just started to put the titers up again, so we will be reinvestigating. So I think this is a sort of example of the fact that there is some hope out there. It's not a hopeless situation. It's become more of a chronic disease for many patients, but we do need that test to allow population screening. Thank you very much.
In the words of Christopher Reeve, actor, author, quadriplegic, and cutting-edge medical research advocate, so many of our dreams seem impossible. Then they seem improbable, and then when we summon the will, they soon become inevitable. Thank you for that introduction. I don't live up to all of that. My focus today is on a 17-year-old passionate personal dream. And that dream is to delete the words impossible and improbable from the ovarian cancer equation and find a test that leads to the early detection of ovarian cancer. I long for the day when this lethal silent killer is permanently consigned to history. Therefore, this presentation is about garnering the will amongst like-minded people to make this happen. Ovarian cancer is the most deadly of all gynecological malignancies. A woman dies from it every 10 hours in Australia. Only 30% of sufferers survive, compared with 60% of those who have breast cancer. Why? Firstly, because sadly over 75% of women are diagnosed at an advanced stage, as we've seen today, dramatically reducing their survival chances. Symptoms are vague and are unable to be detected at an early stage, hence the term silent killer. I'm just going to segue here for a moment. I have found this morning so interesting and so fascinating because I actually put the blinkers on. I didn't want to know about my cancer, so thank you very much. But also, I didn't fit any of the symptoms you saw on the board today. I did. What made me go to the doctor was I was extraordinarily tired, but I might say today I'm still extraordinarily tired. Secondly, research into this insidious disease has not had the support from considerably more consumer voices, as have breast cancer and prostate cancers, which have developed early detection programs. Consequently, there has been limited progress in the early diagnosis of ovarian cancer. The statistics show that 1 out of 80 women will contract the disease. Hence, today, there are thousands of women in Australia walking around unaware that the silent killer is in their bodies, as I was unaware 17 years ago. My own diagnosis was devastating and not very promising. You are alone in your battle, in your thoughts and actions, and you absolutely do not think of the future, as you are fairly sure there is no future. It is a very lonely journey. You are emotionally as well as physically gutted. All dignity is stripped away. It is the lowest you can go. The chemotherapy and other treatments make sure of that, just in case the operation hasn't done its job properly, which, as I said, in most cases, 70% of sufferers, the operation isn't enough. The chemo program is one of the harshest able to be given to a cancer patient. You literally shrink from a vibrant, loving mother and wife to a miserable creature barely able to summon the energy to walk or talk at times. Your family has to watch and endure unbelievable worry, a true regret of mine. I have a daughter sitting in this audience today, supporting me again. Therefore, you can imagine my joy to be told in January this year that Dr. Goli Samimi and her research team at the Garvan were the first in the world to detect DNA methylation changes in the blood that indicate the presence of ovarian cancer. And I repeat, that's at the Garvan Institute and the first in the world to achieve this step. Ladies and gentlemen, this medical breakthrough has the potential to turbocharge the development of an early diagnostic tool for ovarian cancer, thus removing the words impossible and improbable from the ovarian cancer equation. This finding suggests an accurate early diagnosis through a relatively simple blood test is achievable and would greatly improve the survival rates of women given the advantage of early diagnosis. It will turn around the present shocking survival statistics, that is, instead a 30% 5-year survival rate can be changed to a 90% 5-year survival rate with early diagnosis. My job now is to garner the will to render the potential of this cutting-edge research inevitable, which is why I'm standing here today talking publicly about my own ordeal. We need the research like this being done at the Garvan, and we need the public to know and learn about the Garvan's important work. I dedicate this speech today to a friend of mine, a dear friend, who lost her ovarian cancer battle. I held her hand and looked into her eyes and tried with an understanding heart to give her strength in her courageous battle. So many women today are fighting the same battle. Think about it. The woman beside you, your mother, your wife, your daughter, and yes, it's about our granddaughters too. This is why I'm here and why it is imperative we find an early detection program. This is why we need the research teams at the Garvan. Thank God for them, and thank you for listening to my story. Thank you.
Thank you, Rob, for that lovely introduction, and thank you so much for your wonderful speech. I think it really brings home to us why we get up every day and go to work. Yes, today I'm going to tell you a little bit about genomic research in ovarian cancer. When we talk about genomic research, we're going to talk about the genome, so the patient, the woman's own genome, as well as the tumour genome and what has become deranged in the tumour. So we use this genomic research to address a number of questions, to understand what actually causes ovarian cancer, to understand why patients respond so differently to treatment, to develop more effective therapies, and I think very importantly, to select the very best treatment for each individual patient. So I think, as we've heard a little bit about today, cancer in general is not a single disease, which is why it's been so difficult to treat. It's probably more than 200 diseases, all of which have different causes and require different treatments. And ovarian cancer itself, even epithelial ovarian cancer, we're learning is not a single disease. There are different causes and, again, different treatments, different responses to treatment. So I'd like to talk a little bit about cancer risk and what it is that we think causes ovarian cancer. We've had a little bit of a discussion in our earlier talks today, but at the moment we think that risk, and little triangle in the middle there, is a combination of the genes that we inherit, so either the cancer risk genes that Professor Hacker spoke about, or just what we call single nucleotide polymorphisms, or small individual variation in genes. Not changes that would be enough to actually give you cancer, but just enough to lay the foundations or to protect you. So these bottom two are inherited features. And laid over the top of that is what we decide to do with that, so our lifestyle and environment. So our risk of cancer is really a combination of all of those things. And I'm just going to give some examples to make that point a little bit clearer. So let's think, for instance, of skin cancer. There's a little bit of a contribution of inherited genes, but really it's the combination of individual variation of having fair skin with sun exposure. So as you can see, a combination of features that would increase one's risk of getting skin cancer. Similarly, something like lung cancer. Certainly there's individual variation. We know that there are certain SNPs that will actually protect you from, we've all heard about people who've smoked all their lives and never developed cancer. Well, of course, they may have what are called protective SNPs. So just individual variation, small changes that can alter your risk. So what about ovarian cancer? So I've just put up what I think at the moment in terms of ovarian cancer risk. If I put this slide up maybe 5 years ago, it would have looked a little bit different. I would have had these two circles as being quite small because, as Professor Hacker mentioned, we've traditionally thought that BRCA1 and 2, which is genes responsible for having cancer in the family, account for about 10% of the risk. So I would have had this much smaller. I would have had the individual variation smaller, and I would have had the lifestyle as bigger. But in fact, the more we learn, the less confident I am about knowing what the contribution of these different things are. Certainly in the last couple of years, we've identified a number of these genes for which there are just these small variations. I've got a couple of them named here. MERIT40, a couple on chromosome 8, and a gene called TERT. There are a number of these papers have come out of the Australian Ovarian Cancer Study that Rob mentioned and can be found on the website. So certainly there are more of these genes where the risk is, the increased risk is small, but that in combination with lifestyle factors can be enough that may push a woman into actually having ovarian cancer. Professor Hacker's already spoken about lifetime ovulations, hormones, smoking, asbestos diet. All of these things we know may increase risk a little bit. Now, what I'd like to tell you about is some very recent data now that we have on familial cancer, on BRCA1 and 2. As I've mentioned, we've usually thought that was at about 5 or 10%, but we've just completed a very large study as part of the Australian Ovarian Cancer Study where we've comprehensively sequenced BRCA1 and 2 in over 1,000 ovarian cancer patients from Australia so that we can more accurately determine what proportion of patients are carrying this mutation. And this paper is in press and it's about to come out in a journal called the journal JCO, Journal of Clinical Oncology. And what we've found is, in fact, the rate of BRCA1 and 2 mutations are much higher than what we were expecting. So in the general ovarian cancer population, it's up to 13%, but in a group called high-grade serous cancer, so one of the most common types, it's actually over 20% of the cases from patients that have a BRCA1 and 2 mutation. And the important feature is that not all of those patients have got a family history that they know of. And so at the moment, the recommendation for sending women to have testing is whether they have a family history. But we're confident that the results from this study are set to change practice, and then probably it will be all women with epithelial ovarian cancer under the age of 70 will probably be recommended for testing. And the reason we're excited about that is, as Professor Hacker mentioned, we have different treatment options. If we know that a patient is actually carrying one of these mutations, they may be eligible for one of these new drugs, for a PARP inhibitor, which is targeted specifically at women who've got this mutation, and even one of our old friends, the platins, the carboplatin. We know that these patients actually respond really well to some of those drugs. So knowing that a woman might be carrying this mutation may change treatment. And of course for their family it's very important because if it's known that there's a predisposing gene in the family, of course other members of the daughters, sisters, etc. might choose some risk-reducing activities such as taking oral contraceptive pill, as Professor Hacker mentioned, having risk-reducing surgery or targeted monitoring. Now, some other data that's come out just recently is that while we've concentrated very much on BRCA1 and BRCA2 in terms of testing, in fact, there's a small number of lots of other genes that have been found that contribute to familial ovarian cancer. In the past, the way that testing's been done, it's been impossible to test for all of these genes, but these changes in technology means that in fact a lot of the labs are now gearing up to not only look for mutations in BRCA1 and 2, but also for these other members of these what they call BRCA1 and 2 pathways. So these are genes that interact with the BRCA1 and 2 and have a similar effect. So I think that we're sort of excited about those results because, if we can actually reduce the incidence of cancer, of ovarian cancer, moving forward, I think that would be a great advance. So what we've spoken of so far is really what is involved in going from a normal ovarian cell to a cancerous cell. But one of the very important things that we're looking at is what is it that determines how well a patient might respond to treatment. And we've spent a lot of time investigating features of the tumour itself. I've actually just got a little example down here of CA125 plots. Again, we've had lots of mention of that. So this is over time, a patient who has an elevated level to start off with. It's reduced with chemotherapy and surgery, but at some time later, when she relapses, the CA125 goes back up. Then she's responded here, but that's lasted a very short time and she's not responded again. So that's a pretty typical sort of response, if you like, the development, just showing an indication of development of resistance. And so we've been looking at features of the tumour that might be able to help. Now, I've mentioned that ovarian cancer is not one disease. There are probably many. Some types of ovarian cancer, we can actually see that they're different when the pathologist looks down the microscope. So he can see the majority are serous, which we've mentioned a couple of times, but there can be other subtypes. And we know that some of these other subtypes respond a bit differently to treatment. But even within a particular group, so within serous, that looks exactly the same down the microscope, there's an amazing difference in response. We can have some patients that don't even respond in the beginning and others that have a fantastic response to treatment and are essentially cured. But at the moment, we can't predict which patients are going to respond and which ones aren't, and we've got very limited options once that drug resistance has set in. So one way that we've looked at this is we've looked at the gene expression in the tumours to ask, are there gene expression patterns that we can see that might be able to predict. And I'm going to show you a result that's going to appear here that came from an experiment that we did in the Australian Ovarian Cancer Study that Rob mentioned. So in this study, we recruited patients from all the major gynae-oncology centres across the country, including the Women's Hospital here, and Rob and the people here at the Garvan also participated in this. We recruited, the aim had been to recruit 1,000 patients and 1,000 controls, but in fact the patients were so fantastic and the clinicians were so supportive, we overshot the mark and ended up recruiting 1,800 women, so almost doubling our recruitment for this study. So we collected all sorts of information about the women's lifestyle, their diet, we collected tumour samples, blood samples, with as much as information as we could to get the best picture that we could about the patient. And in fact, because it was such a fantastic resource, we've made that available to other researchers. So any other ovarian cancer researchers from around the world can apply. And we've actually supported over 70 different projects to date. So it's actually it's very satisfying to have done this work and to see a lot of other people being able to make the best use of it. But to get back to this experiment, so in this experiment we took samples from just over 250 patients to look genome-wide at what genes were turned on or off, what was expressed and not expressed in these tumours. Now, to give you a little bit of put this into perspective, at the time we did this, the next largest study in the literature was about 80 patients, and the list price for doing the assay was $1,000 a sample. So that just gives you a little bit of an idea of how big this experiment was. We took retrospective cases from our tissue bank at Westmead, as well as cases from the Australian Ovarian Cancer Study. And what you will see, similar to what Goli showed earlier, is that where you see a pattern of green, it means the gene's low, and where you see a pattern of red, it's high. So this here, in one snapshot, is a result of almost 9,000 genes in 251 tumours. So what we've done here is we've asked the computer, so this is like a grid, so the tumours are going in this direction, so they're lined up in this direction, and going across this way are individual genes. And where you see a pattern of red, it means the expression of those genes in those tumours are high, and where you see a pattern of green, it means these genes in these tumours are low. So we've asked the computer to find the list of genes that best segregates the tumours into different groups, and what groups, ask the computer to put them into the groups. And as you can see, those patterns are pretty clear. Now the important thing is, what I've done here is I've blocked out 3 and 6, because in fact they're two groups that just looking down the microscope you could tell that they're going to be different. Okay, so we already know what's underlying that difference. But in fact, 1, 2, 4 and 5, they all look the same down the microscope. So to the clinician, they would not know that there was any difference in the patients. But as you can see, when we look down at the gene expression level, they look really different. And not only that, if we look at these patients here in what we call cluster 1, they've got very high expression of this cluster of genes here, they actually do poorly. Whereas the patients in 2 and 4, so this group here, 2 and 4, actually have a much better survival. So those gene patterns can actually predict whether a group of patients might do well or not. The problem is that it's not absolute, like many of these things, because within one, even though in general they do badly, there are a couple of patients who do really well. So just this is not going to be enough. But what it might do is that if we find the pathways that are activated in these different subtypes, then we might be able to either select drugs or find drugs that might target just the patients in those pathways. So what we've been able to do, so this again is those results repeated in this slide. What we've been able to do is to actually look at other people's data. So since we did this study, there was a big study undertaken in the United States where now they've gone even higher, over doubled our number. And we were very pleased to see that when we analyse their data, we get these same 4 groups. And similarly with another data set from the NCI. So it's not as if, because that's been one of the problems with these things, is that we might get one result, but then we might not be able to repeat it. So it's been very pleasing to see that you can get those same results in completely different patients. And similarly, the people at the TCGA, when they did the experiment completely independently, they also found 4 groups, and when they reanalysed our data, they also found that same information. So it's pretty clear that these serous tumours, there are at least 4 particular, 4 specific molecular groups. But as I've said, what we really need to do is find the best treatment for each subtype, and then ask, are there other predictors of outcomes? Because we know that this is not the whole story. And one of the things that we've done is we've been lucky enough to be part of what's called the International Cancer Genome Consortium. So this is a consortium where we're aiming to fully sequence the whole genome of 50 different cancer types and 500 specimens of each different cancer type. And just to put that into perspective, it took 13 years to do the whole genome of one person. And this is going to be doing the whole genome sequencing of 25,000 genomes. So just to show how technology has moved ahead, that we'll be able to do this type of work. So just under here is pancreatic cancer, and of course that's a study that's from the Garvan. But so our contribution is going to be ovarian cancer, and we're going to be looking specifically at mutations that are involved in drug resistance. Oh, and I just wanted to mention the other exciting thing about that study is that the results are put straight onto the web, so that in fact other researchers will be able to go on and interrogate that data and ask their own questions. So we've talked a lot about what might be happening in the actual tumour itself, and that's where many of us have spent our careers looking at the features of the tumour. But more recently, we've spent some time looking at genetic variation in the woman herself, in the way that a woman might handle the particular chemotherapy, for instance. And we know that some women have terrible side effects, others don't, some respond well, some don't. Is there a component of that that's actually inherited? So the way that our bodies might eliminate the drug, for instance, is that one of these small SNPs or small genetic variations. And so we're doing some work now looking at genetic variation and how that can affect drug metabolizing and transport genes, and how that may in turn actually contribute to how well a patient might respond to treatment. And so this is actually called, it's got a whole new name, pharmacogenomics, or the genetic variation and response to treatment. So in other words, we've got patients here, a group of patients that might all have the same condition, but using DNA profiling, we might be able to split them into groups that have got a good response, a poor response, or those that have very bad side effects. So what we've done in the Australian ovarian cancer study is that we've taken a group of patients that have had an incredibly good response, those that have had a very poor response, and we've looked for these SNPs or small genetic changes across the genome. And I've just put an example of that up here. So this work was done in collaboration with Georgia Shevnik's-Trench from the Queensland Institute for Medical Research. So what we've lined up here, this is actually called a Manhattan plot, because it looks like the skyscrapers in Manhattan. And all along the bottom here, we have each of the chromosomes all lined up. So chromosome 1, 2, 3, all the way through here to 22. And on this axis, what we actually have is the probability. So this is a probability that under this peak, there's a difference between these groups. So here, for instance, we've got these little dots that go up really high. And so that means that they've got what's called a good p-value. And so the probability that under that peak, in this area of the chromosome, there lie genes that are different between the patients who've got a good response and a poor response. And the reason is this is important is that it might help us to then identify the best treatment for a particular patient. So we're pretty excited about those results. Now, the last aspect that I'd like to tell you about today is, so we've looked at the tumour itself, the whole patient. Now, what about their lifestyle? Are there changes? Are there things that that patients do once they're diagnosed? They might change their diet, they might exercise, or they might not. And how do these things affect their outcome, or can they affect their outcome? So we've just started a brand new study called OPAL, and this is led by Penny Webb, again at QIMR. We've just had the funding for this. We've got ethics through at most centres. Again, this will be also the Royal Women's Hospital, and Professor Hacker will be participating in this. And we've had our very first patient recruited in Tasmania, so we're pretty excited. And so here we're going to ask those questions. What features of lifestyle, diet, other medications, etc., might actually impinge on how well the patient responds to treatment. So overall, I hope that I've convinced you that ovarian tumours are not all the same, and the patients are not all the same, and we're trying to use genomics to sort these patients into their like groups so that we can better understand risk and prevention, so that we can optimise treatment so that the patients get the best treatment for their particular cancer, and to optimise and develop new targeted treatments. I'd just like to thank our patients who've all been absolutely fantastic in these studies. The whole AOCS study group, which is led by David Botel down at the Peter MacCallum Cancer Centre, and this is his group, and Georgia Shevnik's-Trench and Penny Webb also on the management committee of this study, and their group at Queensland, my group out at Westmead, and of course all of our funders. Thank you.
Good morning, everybody. This is a word from your sponsor. It's my great privilege to work for the Garvan Research Foundation. We're like the sales and marketing arm of the Institute, and it is an incredibly exciting place to be working. Just before I tell you a little bit about what we're actually doing, I thought I'd show you our new CSA, or Community Service Announcement. It's really a commercial, but we don't have to pay for it when it goes to air. And you'll be seeing this sometimes on television, so I thought I'd show it to you. It's just being produced.
A cure for cancer, for diabetes, for Alzheimer's? Our generation can do it. Today we're in the midst of a revolution in gene technology. And at the Garvan, we're working hard with these technologies to find new treatments for a whole range of diseases. With your help, this will be the last generation to die from cancer. Today we really believe cures are within reach. All that's holding us back are the resources to attack the problem. Finding the answers depends on the financial support of people like you. Cures are within reach.
What do you think? Gives you a great spirit of hope, doesn't it, that we're getting very close to answers to many of these debilitating diseases. At the Garvan Research Foundation and at the Institute, of course, we have a fantastic history of research, not only in Australia but around the world. But what many people don't realise is that for every dollar that we receive from the government, the foundation actually has to find an additional 70 cents so that our staff have access to the latest and best infrastructure, equipment and facilities so that we're able to attract the best and brightest minds from around the world, which we've been able to do. Donors to the Garvan Research Foundation are really making sure that we can improve the health outcomes not only in Australia but in the world at large. So we've been greatly helped by the community, by people like yourselves. And I'll just give you a little overview of the things that we get help with, and maybe there are some areas there that you might be encouraged to help us with as well. Got that to move. It's exciting, isn't it? Next time I'll have sound effects. We do get some wonderful support from our corporate partners, and in fact today we've got some lovely volunteers from MLC who've come to help us to put on today. So they're one of our corporate sponsors, and you've actually seen some of them listed earlier when Goli was talking about the support that we get. Word of mouth is very important, so all you people here today are wonderful ambassadors for our work. So please feel free to go and tell everybody about the work that's happening here at the Garvan and how it's making such a difference to everybody's health outcomes. And talking of word of mouth, this is actually going to be webcast today, so people are able to watch it at home, in privacy of their own home. And I hope if you're watching it that you'll be encouraged to have a look on our website and make a donation as well. So donations are incredibly important to continue our work. And you may have noticed that we're coming up to the end of the financial year. We've certainly noticed. Thankfully, we've got wonderful donors who have, opened up their checkbooks or their credit cards, and if you come up to the foundation, you'll see that there's quite an amount of donations coming in. So we're very, very grateful for the support that we get in that way, particularly at this time of the year. A community fundraising, you're all able to join up and go on the City to Surf, or you can run it, you can walk it, you could even have a cup of tea if you wanted to, and just have a high tea and raise funds for the Garvan. I'm thinking cup of tea might be more my thing. And we have a number of workplace giving programs in corporations and companies where people are able to actually give a donation pre-tax, which makes it go a bit further. And finally, I'd just like to mention an area that's very dear to my heart, as I'm the bequest officer here at the Garvan. And many of our programs are actually up and running today because of the wonderful generosity of previous generations leaving a gift in their will. So if that's something that may be of interest to you, I hope you will think about it when the time is right. And it doesn't matter if it's large or small, because every gift in your will does make a huge difference to our work. So if you'd like to come out and see me at morning tea, I've got the correct wording for you, Will. Or if there's anything else you'd like to learn about that you think you'd be encouraged to help us with, we'd be very, very grateful. So I hope you enjoy your cup of tea. Please come over and say hi.